This video will focus on some basic concepts related to gas exchange and circulation. And although these concepts can be applied to virtually all animals, we will focus primarily on gas exchange and circulation in human beings. To begin with, let's just review some basic concepts about gas exchange. There is a simple equation that we could look at, referred to as Fick's law of diffusion. Now, the equation itself isn't all that important, but what is important is essentially are the outcomes of this basic equation. So Fick's law of diffusion basically outlines some conditions that should be met in order to maximize the rate at which gas exchange takes place. And these should all make intuitive sense if you just think about it. So for example, it basically states that the larger the surface area of a gas exchange organ, the more likely gas exchange is to take place. So imagine the inner lining of your lungs, tremendous amount of surface area where oxygen is diffusing into your bloodstream, CO2 is diffusing out, and again, the bottom line is the larger the surface area, the more gas exchange will take place. Also, the thinner the respiratory surface across which gas exchange takes place. So the thinner the surface, of course, the more easy it will be for gas exchange to occur. And also the higher the pressure gradient, the more likely gas exchange is to take place. So if you compare the partial pressure of CO2 in the blood in your lungs, that's going to be higher than the partial pressure of CO2 in the air that you inhale or breathe in. So as a result, CO2 will be more likely to diffuse out of your lungs than in. So again, the higher the gradient, the more likely gas exchange is to take place. If we focus on gas exchange organs in humans and other mammals and many other vertebrates, we of course are talking about lungs. Lungs are these specialized organs, of course, suspended in our chest cavity. The functional unit of lungs are these sac-like structures we refer to as alveoli. So thousands and thousands of alveoli within our lungs. Notice these alveoli are loaded. They're just totally permeated with lots of capillaries. So lots of things taking place in the capillaries in our lungs. Of course, oxygen making its way into the blood in those capillaries, and CO2 making its way out. How then do lungs function? How do we breathe? Lungs themselves don't really do a whole lot. They don't play a very active role in the breathing process. They're essentially these organs that hang suspended in our chest cavities. And when we breathe in, essentially what happens is we contract our diaphragm, this muscle at the base of our chest cavity, as well as some muscles between our ribs. That essentially increases the volume inside our chest cavity. That creates a partial vacuum, some negative pressure in there, and of course the lungs simply respond by inflating and pulling air in, of course, through our nose or our mouth each time we inhale. So again, lungs themselves play a fairly passive role. They just change in response to pressures within our chest cavity. Let's talk briefly about some of the basic types of blood vessels in our bodies, focusing initially on arteries and veins. The main functional difference between arteries and veins, arteries are those blood vessels that carry blood away from our heart, and veins are those blood vessels that carry blood back toward our heart. They also have some structural differences. Arteries tend to have a fairly thick muscular wall to them, so they actually contract, and they play a large role in maintaining blood pressure within your bodies. Veins, on the other hand, tend to be fairly thin-walled, and the blood pressure within veins tends to be quite low, and although there aren't any illustrated in this image here, veins often have valves, which help to prevent the backflow of blood as it's making its way back toward the heart. Probably some of the most important types of blood vessels in our bodies are capillaries. Capillaries are, of course, very thin, very small, narrow diameter blood vessels, not much bigger than individual red blood cells. But this, of course, is the business end of your circulatory system. This is where a lot of the exchange of nutrients, oxygen, CO2, and so forth is taking place between your bloodstream as well as all of the surrounding cells throughout your body. So let's take just a minute and review the pathway of blood flow through a typical four-chambered heart like we find in human beings. We could begin this pathway anywhere, of course, but let's just begin with deoxygenated blood that's just returning from various parts of our body, returning back to the heart. It it returns back to the heart through this big blood vessel referred to as the vena cava. Now you could distinguish the superior vena cava and the inferior vena cava, but the bottom line is the vena cava carries deoxygenated blood back to the heart and dumps it into the right atrium. And so the right atrium, labeled B in this image, fills with blood. It then contracts, forces that deoxygenated blood down into the right ventricle, and on its way it passes through this valve right here, labeled C, which consists of 3 flaps of tissues. Therefore, we call this valve the tricuspid valve. When the right ventricle contracts, it then forces the blood up through this major blood vessel here, labeled E. This is the pulmonary artery. Although it's carrying deoxygenated blood, it is considered an artery since it's carrying blood away from the heart. And although it's not labeled, there are valves within the pulmonary artery that are essentially referred to as semilunar valves. So from the pulmonary artery, deoxygenated blood makes its way to the lungs. After it goes to the lungs, of course, it gives off its CO2, picks up oxygen, and it makes its way back to the heart via the pulmonary veins, labeled F in this image. So the pulmonary veins carry oxygenated blood back to the heart. The blood then collects in the left atrium, labeled G. Left atrium then contracts, forces blood down into the left ventricle, labeled I, and of course passing through this valve right here, labeled H, which consists of 2 flaps of tissue. So we refer to this as either the bicuspid or the mitral valve. So the left ventricle contracts, forces blood out through this major blood vessel here labeled J. This, of course, is our aorta, which carries oxygenated blood throughout most of our body. So the blood then goes throughout our body, gives up its oxygen, picks up CO2, makes its way back to the heart via the vena cava, thus completing the cycle. So if you think about this pathway of blood flow through a human heart, we can essentially distinguish 2 circuits. On one hand, we can talk about the pulmonary circuit. This is when blood, which is pumped by the right ventricle, makes its way from the heart to the lungs and then back to the heart. On the other hand, we could talk about the systemic circuit. This is where blood is pumped from the left ventricle to most of the body, and that blood, of course, returns back to the heart. So these 2 circuits, pulmonary and systemic, involved in circulation within a 4-chamber heart. And whether you're talking about blood flowing through the pulmonary circuit or the systemic circuit, of course, blood is eventually going to be passing through lots and lots of capillaries, whether they be in your lungs or throughout the rest of your body. And so there's some important implications as blood flows through capillaries. So we're going to address this basic question, that is, what happens to blood, especially blood pressure and velocity, as it travels through capillaries. So take a look at this image here, which illustrates some fairly important relationships. Let's focus on this bottom panel to begin with. What we're looking at here is blood flow as it's flowing through arteries, beginning with major arteries, then smaller ones, and eventually through capillaries. And then that blood then makes its way through small veins, and then larger veins, of course, as it makes its way back to the heart. So let's focus initially on this bottom line in this graph right here. What this line represents is total cross-sectional area of these different blood vessels within your body. So if you looked at the total cross-sectional area of your aorta, and then smaller arteries, and then eventually capillaries, and then small veins, and then larger veins, vena cava, what we see is this relationship where the capillaries have a lot more total cross-sectional area than do all of the other blood vessels in your body. How does that relate to the speed or the velocity at which blood is moving? Well, if you take a look at this top line here, what this indicates is that as blood moves through major arteries, it's traveling quite fast. But when it makes its way through all those capillaries, it's traveling quite slow. As the blood makes its way back to the veins, again, the speed picks up, and so velocity is quite high. But the bottom line is blood is moving fairly slow as it's traveling through capillaries. What about blood pressure? Let's take a look at the top panel here. What we see is that blood pressure is quite high to begin with. But then as that blood makes its way through capillaries, that blood pressure drops off dramatically. And you'll notice that pressure is never regained. So as blood is moving through veins on its way back to the heart, it is moving under fairly low pressure. So the bottom line is, as blood is moving through capillaries, not only is the speed at which it's traveling quite slow, but it's also under fairly low pressure. So let's now take a quick look at this process of capillary exchange. In other words, as blood is moving through capillaries, how does that fluid portion of blood and everything dissolved in that fluid portion, whether it be oxygen or nutrients and vitamins, how does that fluid portion or plasma exit the capillary on one end and re-enter the capillary on the other end? So again, the basic process is referred to as capillary exchange. And this whole process is illustrated in this image below. So blood is moving through this capillary from left to right. As blood first enters the capillary at the arterial end, eventually that blood will make its way to the end of the capillary here at the venous end. So as blood is moving through the capillary, there are two important forces that we should take note of. On one hand, there's blood pressure. Not a whole lot, but there is some blood pressure. Ultimately, that blood pressure tends to force fluid out of a capillary. But on the other hand, there's also an osmotic pressure. So as long as the concentration of solutes in blood is higher than the concentration of solutes in the surrounding tissues, the osmotic pressure will tend to draw fluid into a capillary. So these two opposing forces at work throughout the capillary. So what you'll notice is that when blood first enters a capillary here at the arterial end, notice the blood pressure exceeds osmotic pressure. As a result, fluid tends to leave the capillary. But as blood makes its way through the capillary, if it's losing fluid, what's going to happen to the blood pressure? Well, the blood pressure will continue to drop. And so that by the time blood exits a capillary, the blood pressure is less than osmotic pressure. So here at the venous end of a capillary, the net result is most of the fluid tends to return back into the capillary before that blood makes its way to veins and eventually makes its way back to the heart. And so ultimately it's this loss of fluid and the regaining of most of that fluid that essentially is responsible for bathing all of the cells in your body with the important components that we find within our bloodstream. Now, most of the fluid does return back to a capillary. The fluid that does not make its way back into the capillary is ultimately picked up by your lymph system and eventually is returned back to your circulatory system. And so now let's just spend a minute discussing briefly how does the heart work. We refer to the heart as being myogenic. This just simply refers to the fact that your heart has the ability to produce its own electrical impulse, which causes the atria and the ventricles to contract. And we can, of course, pick up this electrical activity of each heartbeat in the form of an electrocardiogram, or EKG or ECG, illustrated in this image here. So if we take a look at a single heartbeat, let's begin with pointing out there's a special little patch of cells up in the right atrium referred to as the SA node or the sinoatrial node. So the SA node, this sort of serves as the pacemaker for each heartbeat. This generates the impulse, which spreads throughout both atria, causing them to contract. Then that impulse makes its way to another patch of cells called the AV or the atrioventricular node, which essentially delays that impulse for a split second, long enough for the ventricles to fill with blood. The AV node then sends the impulse down through the ventricles, causing the ventricles to contract in an upward-like fashion, pushing blood, of course, out of the two major blood vessels, namely the aorta and pulmonary artery. Then the ventricles relax, thus completing a single heartbeat. We can, of course, pick up the activity of a single heartbeat, much like you see here. So this first little blip right here, referred to as the P wave, this, of course, is the impulse causing the atria to contract. This next major wave here is our QRS wave or QRS complex. This is electrical impulse causing the ventricles to contract. And then this final wave of activity toward the right here, this is the T wave. This essentially represents the relaxation of the ventricles. So of course ECGs can be used in many different ways to help diagnose various types of heart problems. So let's spend just a minute talking about hemoglobin. We all know that one of the major roles of blood is to carry oxygen throughout all of the tissues within our body. So how is this accomplished? Well, hemoglobin is this protein which consists of 4 separate chains of amino acids. So here we see a single hemoglobin molecule illustrated. And of course, hemoglobin has this remarkable ability to either pick up oxygen or release oxygen depending on the conditions where it is found. So because hemoglobin consists of 1, 2, 3, 4 chains of amino acids, a single hemoglobin can hold 1, 2, 3, or 4 molecules of oxygen. And so let's take a look at the ability of hemoglobin to either pick up oxygen or release oxygen in the form of an oxygen dissociation curve, and we'll also illustrate what's referred to as the Bohr shift. So let's begin by taking a look at this image here. And so this is simply an oxygen dissociation curve which represents blood at different partial pressures of oxygen on the x-axis compared to the amount or the percent of hemoglobin that's totally saturated with oxygen. In other words, the percent of hemoglobin that's bound or holding on to 4 molecules of oxygen. So notice what we see here is that, as you might guess, as hemoglobin is found in increasingly higher and higher partial pressures of oxygen, it's more likely to hold on to or to bind to the maximum amount of oxygen as possible. And so that's essentially what's illustrated in this figure. And so how do we interpret a figure like this? Well, let's just take a look at a few questions. We can ask, first of all, in lungs, what percent of hemoglobin is totally saturated with oxygen? Well, you see, right about 100%. We might also ask, okay, at rest, what percent of the hemoglobin is saturated with oxygen? Well, if you're just sitting there not doing a whole lot, you might say, well, at tissues at rest, the partial pressure of oxygen is about 40 millimeters of mercury. At that partial pressure of O2, looks like about 50% of hemoglobin is saturated with as much oxygen as possible. So given that, we can then address the basic question, okay, what percentage of oxygen is released to tissues at rest, and then how about during exercise? Well, we said at rest, about 50% of the hemoglobin is saturated, therefore 50% of the oxygen is released to surrounding tissues. What about during exercise? Well, you're using up oxygen during exercise, so the partial pressure of O2 in those tissues during exercise is lower. If we look at the percent of hemoglobin that's saturated in those tissues during exercise, we could say, well, about 20% of hemoglobin is saturated, therefore about 80% of the oxygen is released from hemoglobin during tissues, into tissues during exercise. Okay? So that's essentially how you interpret a figure like this. Then we can also take a look and say, okay, beyond just the partial pressure of oxygen in the surrounding environment of hemoglobin, hemoglobin is a protein. So its activity is in part determined by environmental factors such as temperature or pH. And so what we can do is address this basic question, okay, if you exercise and you produce a little more CO2 in your tissues, that's going to decrease the pH of your blood just a bit. What effect does that have on the properties of hemoglobin? And so that effect is illustrated here in what's referred to as the Bohr shift, simply named after the person who first observed this. So at rest, the pH of your blood is about 7.4, and here we see the oxygen dissociation curve at that pH. If you exercise, again, the pH of your blood drops just a bit, but that drop in pH, just that slight acidic environment compared to the pH at rest, that drop in pH shifts the whole oxygen dissociation curve of hemoglobin. To the right just a bit, what are the implications? Well, the implications are essentially that at any given partial pressure of O2, and we can take a look at 30 just because if that's the PO2 in tissues during exercise, essentially what that means is hemoglobin is releasing even more of its oxygen than it would if you were just sitting there at rest. So the bottom line is that hemoglobin, in response to a slight drop in pH, is likely to release even more oxygen to tissues in your body, and of course that's exactly when they need it most, right? At times of strenuous exercise. So this Bohr shift we see illustrated here in response to a drop in pH. We also see the exact same shift if we see a slight increase in temperature, which again would occur during times of strenuous exercise. So we've been focusing on oxygen. One final brief point that I'll make relates to the transport of carbon dioxide in our bloodstream. Imagine producing lots of CO2 as a result of cellular respiration at different parts of your body. That CO2 then diffuses into your bloodstream, picked up by some red blood cells. And it turns out that once the CO2 makes its way into the red blood cells, it's converted into bicarbonate ions. And so we see that reaction here catalyzed by this enzyme carbonic anhydrase. And so bicarbonate ions, that's this HCO3-, they're produced, and this essentially is the form in which CO2 is transported in your bloodstream. And notice if the enzyme runs the reaction left to right, that's going to reduce the CO2 in your bloodstream, which of course will increase the gradient compared to the CO2 in your tissues, which constantly favors diffusion of CO2 from your tissues into your bloodstream. Once this blood makes its way to your lungs, the reaction essentially occurs in reverse. And so the bicarbonate ions are converted back to CO2. CO2 diffuses out of the capillaries into the alveoli in your lungs, and of course, we exhale the CO2 at that point. So that was quick, but that gives you a brief